here are the answers for the questions for the critical thinking practice for Chapter 6. Basically, you have a scenario in which we'll say that all of the bacteria have been removed from my teeth and they've been recolonized. And we have bacteria undergoing binary fission every 20 minutes. So now we're assuming that I have 1,000 bacteria per tooth to start. So now I'm asking you to figure out how many bacteria for just one tooth per tooth do I have after 12 hours. So you may look at this problem and have no idea where to start. So let me show you how to do this problem. So the first thing that we need to do is figure out how many generation times we have. It's been 12 hours and the bacteria are dividing every 20 minutes. So how many 20-minute periods do we have in an hour? Well, there are 60 minutes in an hour. We take 60 minutes, divide it by 20, we get 3. So then we take those 3 generations per hour, times it by the number of hours we have, and we get 36 generations per 12 hours. So this is the number of times that the population of bacteria divide during this 12-hour period. So we take 1,000 bacteria times 2, because every time they divide you get 2 bacteria. Each cell produces 2 bacteria. So for the first 20-minute division, we get 2,000 bacteria. We would do this 36 times, because for every generation we're going to have a doubling of the population. And in the end, the answer is 6.8 times 10 to the 13th. That's a whole lot of bacteria. So now I ask you what may seem like a really random question about human generations since the founding of our country in 1776. Or at least that's when the Declaration of Independence was signed. I promise I tie this into bacteria. So how do we figure this one out? This one's relatively easy. We take 2014, subtract 1776 from it, we get 238 years. We divide the 238 years by 20 years, which is 20 years per generation, and we get 11.9 generations. So the question is, am I going to go with 11 or 12? Well, I did this one on purpose because we haven't completed a full generation yet. So you have to drop the fraction. I know, it should be, once it's 0.5, you round up. Well, when we're talking about reproduction, you have to round down. So the answer would be 11. Now, I have my next random question. If you convert the bacterial generations to human generations, so 20 minutes equals 20 years, I'm beginning to bring this back to bacteria. So you do the math and you find out that it's 220 years, or since 1293 AD, or in Europe right before the plague hit in 1300. The reason why I do this is I want you to think about a couple of things. One, how does this change your perspective on brushing? Does it make you want to brush only once a day? Because, then, you have in the 1400 years, instead of 720 years, you want to brush your teeth at least twice a day. Biofilms in general. These biofilms, if we were to convert bacteria years into human years, are really long-standing civilizations. And so it explains some of what's going on when we talk about biofilms. Also, later on, when we talk about mutations or change occurring in bacterial populations, 12 hours is a long time for a population of bacteria. It makes sense as to why, say, bacterial resistance can show up in what to humans seems like a really short time. Things to keep in mind as we move through the course. Okay, on to our next scenario. You've inoculated 100 facultatively anaerobic cells onto nutrient agar. Then you incubate the plate aerobically. Then you inoculate the same number of cells of the same species, so you know they're facultative anaerobes, onto nutrient agar, so same agar, and you incubate the second plate anaerobically, or without oxygen. With optimal growth conditions, this bacterial species will divide every 20 minutes. After incubation for 47 and a half hours, which plate should have more colonies? Now you may be wondering why I picked 47 and a half hours. That's approximately how long your plates are sitting in the incubator for lab. So, here's things to consider. What does facultatively anaerobic mean? Well, it means that the cells can grow with or without oxygen, and they grow better in oxygen conditions. In the next chapter, you'll find out why. Basically, they can make more ATP, generate more energy from their food when they're in the presence of oxygen. So, I look at my answers, options. The anaerobically incubated plate will have more. Well, that's a possibility. They will grow faster because they're facultative anaerobes. The anaerobically incubated plate, okay, both plates should have the same number of colonies, and after 48 hours, the colonies will be too small to see. Well, if they're dividing every 20 minutes, that's not going to be an option, so it's not going to be option D. You have the choice of the aerobically incubated plate, the anaerobically incubated plate, or they're going to have the same number. Well, the answer is you're going to have the same number of colonies. Here's why. These cells can live with oxygen or without oxygen. So none of the cells are going to die. So you're going to have the same number of colonies on the plate. The cells have been spread out, they've been stuck on the agar, and they proceed to divide and produce colonies that we can see. They can't get up and move off of the agar. They're stuck in that one spot. So both plates are going to have the same number of colonies. Now on to my follow-up question. Which plate should have bigger colonies? Well, we know that you're going to have the same number of colonies, but which ones are going to have bigger? Well, it's going to be the plate that enables the bacteria to divide more often. And the cells are going to divide more often in the aerobically incubated plate. They're going to divide more often, you're going to generate more cells, you're going to have a bigger colony. Now, I should warn you, I love to ask a variation of this question on the exam, so let's back up. Suppose that I change this to you inoculated 100 obligate anaerobic bacteria. So if they're obligate anaerobic, that means oxygen is going to kill them. So we're going to have more plates on the anaerobically incubated plates because there's not going to be any colonies on the aerobically incubated plate. See how that works? Then on the next one, the answer is the anaerobically incubated plate because there are no colonies on the aerobically incubated plate. See how that works? So, be ready for this question when it shows up on the exam. Okay, next scenario. I want you to take this disease scenario and I want you to graph the disease progression. So, let's take a look at how I would do it. First, I would do a growth curve where I have a lag phase. This is where, if we were, let's go with the growth curve as it happens in a test tube. So I've just moved bacteria to the lag phase, and basically the bacteria are getting used to their new environment. They may have been in a tube where there wasn't a lot of food, there was an increase of waste, and so they had downregulated their metabolism. So now that they're over here in new fresh media, they've got to turn genes back on and they got to get enzymes going to make use of the food in the new home. Then, here in this spot where we have a rapid increase in numbers of cells, we have the log phase. So I've got cells rapidly dividing, I've got very few dead cells. Then here, as food starts to run out and waste products start to build up, I have what's called the stationary phase. I've got equal numbers of cells dividing, living, and equal numbers of cells dying. Then, if the food starts getting really short supply and I've got more and more waste, then I have what is called the decline phase, where basically I have more dead cells than live cells. So we can take this same growth curve and apply it to a pathogen. Okay, so a pathogen comes in, getting used to your body, starting to turn on genes to make use of you and cause disease. Now when we get to the chapter on disease progression, we'll find out that this is incubation. We don't know about that from the previous scenario. But as the numbers grow, your immune system and your body start to realize that there's something not right. So that's the scratch, the tickle in the back of your throat. Then as you start to sneeze and cough and ache all over, that could be when you've got either high numbers of pathogen at the top of the log phase, or you could move it over here into the stationary phase as your immune system kicks in and starts killing off the microbes. Then as you get better, the numbers of microbes are dropping, you're feeling better, and that's how I would graph it. Okay, so follow-up question. During your illness, you decide to swab your nose and throat to see what is causing your illness. You do a series of pore plates, which is a way of doing a direct assessment of the numbers of microbes, but you only culture normal levels of common human mucous membrane inhabitants. Are they causing your illness? If not, why were you unable to culture the pathogen? So let's go to the answer options, which didn't fit on the first slide. So the mucous membrane inhabitants are causing the disease. Well, I would say this was unlikely. If you've got normal numbers, normal mucosal membrane inhabitants, probably not causing the disease. We're looking for a change. So the pathogen is susceptible to heat and was killed during the pore plate process. That's a possibility. A lot of pathogens are susceptible to variations in heat, but would they still be around when you start to get achy all over, possibly have a fever? It's a possibility, but it's a low possibility. You didn't have the correct media for culturing the pathogen. That's what this chapter is all about. That's a possibility. And the pathogen is a virus and can't be grown on non-living media. We cover that in this chapter also. If you remember from chapter 5, viruses have got to have living media. With your pore plate, all the food is dead. So, just because I know that most colds are caused by viruses, I'm going to go with that answer. But these two are pretty good answers. Well, that's it for this chapter. And let me know if you have questions.